and welcome to some biology class. Friends, today we shall discuss how nitrate is assimilated by plants into organic compounds. Nitrate is a form of inorganic nitrogen available in the soil. Soil sources of nitrate include decomposing plant residues, manures, chemical fertilizers, rainfall and lightning. Plant roots actively absorb nitrate from soil solution via several low affinity nitrate proton co-transporters. The absorbed nitrate is eventually assimilated into organic nitrogen compounds. The first step of nitrate assimilation is the reduction of nitrate into nitrite, and this is followed by further reduction of nitrite into ammonium, and finally ammonium is assimilated into amino acids. Now let us discuss about these individual steps in detail. The first step of nitrate assimilation is the reduction of nitrate into nitrite in the cytosol by the action of enzyme nitrate reductase. In this reaction, NADH or NADPH can be the electron donor. This reaction involves the transfer of two electrons. The most common form of nitrate reductase uses only NADH as the electron donor, whereas another form, which is predominantly found in non-green tissues such as the root, can use either NADH or NADPH. The second step of nitrate assimilation is the reduction of nitrite into ammonium. Nitrite is a highly reactive and potentially toxic ion. Plant cells immediately transport nitrite generated by the action of nitrate reductase from cytosol to chloroplast in leaves and plastids in roots. This reaction, which is catalyzed by the enzyme nitrite reductase, involves the transfer of 6 electrons. Ferredoxin is the electron donor. In this reaction step, electrons are transferred from reduced ferredoxin to nitrite. As a result, ferredoxin becomes oxidized and nitrite becomes reduced to ammonium. Chloroplast and root plastids contain different forms of nitrite reductase. However, both of them consist of a single polypeptide chain which contains two prosthetic groups. One prosthetic group is the iron sulfur cluster and the other one is a specialized heme. Next step is ammonium assimilation, in which the nitrogen in ammonium is converted into the amide nitrogen of amino acid glutamine. Plant cells avoid ammonium toxicity by rapidly converting ammonium generated by nitrate assimilation or photorespiration. The primary pathway for this conversion involves the sequential action of enzymes glutamine synthetase and glutamate synthase. Glutamine synthetase combines ammonium with glutamate to form glutamine. Energy for this reaction comes from the hydrolysis of ATP, and divalent cations such as magnesium, manganese or cobalt serve as cofactors. Plants contain two classes of glutamine synthetase. One is in cytosol and the other one is in root plastids and shoot chloroplast. Elevated plastid level of glutamine stimulates the activity of glutamate synthase. Glutamate synthase catalyzes the transfer of amide group from glutamine to 2-oxoglutarate to form two molecules of glutamate. Plants contain two types of glutamate synthase. One accepts electrons from NADH and the other one from reduced ferredoxin. Ammonium can also be assimilated by an alternative pathway in which the enzyme glutamate dehydrogenase deaminate or synthesizes glutamate. After its assimilation into glutamine or glutamate, the nitrogen is also incorporated into other amino acids via transamination reactions. The enzymes that catalyze transaminations are called transaminases or aminotransferases. For example, aspartate aminotransferase catalyzes the transfer of amino group from glutamate to oxaloacetate to form aspartate and 2-oxobutyrate. All aminotransferases require pyridoxal phosphate as cofactor. Amino acids asparagine and glutamine link carbon and nitrogen metabolism. Asparagine is not only a component of proteins but also serves as a key compound in the transport and storage of nitrogen. This is because of its stability and high nitrogen to carbon ratio. The major pathway of asparagine synthesis is the transfer of amide group from glutamine to aspartate. The synthesis of asparagine from aspartate involves the hydrolysis of ATP into AMP and inorganic pyrophosphate. Humans and most animals cannot synthesize certain amino acids such as leucine, isoleucine, lysine, histidine, phenylalanine, tryptophan, valine and methionine. By contrast, plants can synthesize all the 20 amino acids which are common in proteins. The nitrogen-containing amino group for amino acid synthesis is derived from transamination reaction, and the carbon skeleton comes from various intermediates of glycolysis such as 3-phosphoglycerate, phosphoenol pyruvate and certain intermediates of the citric acid cycle such as oxaloacetate and 2-oxobutyrate. So friends, this is all about nitrate and ammonium assimilation. For more updates, please subscribe my YouTube channel, Sam Biology Class.